Okay, so the paper we're discussing today is modular design of a synthetic payload delivery device, and it's by Christopher Anderson and others. The reason the paper was because not only is it a synthetic system that can be optimized module by module. Can you stop recording for a second? Are we recording?
Yes, we are.
All right. Sorry. I might not be able to do that, the whole thing. Hello?
Yeah.
Yeah.
Jump in.
Yeah.
Start off.
Can I cancel the whole thing?
Can you what?
Hello?
What's up, Yuriy?
Cancel it on rain check? No?
Yeah, don't stress.
It's a very casual thing.
Yeah, you won't be tested on it, Yuriy. Well, maybe I'll test you.
Okay.
Yeah, probably so. That's
Would you like to
Well,
would you like to
yeah.
to start off with anything, or would you like someone else to try to walk through it a little bit? I think you can.
Whichever's what?
Why don't you keep going? Because I know you put a lot of work into this, and I think
Yuri, try turning your muting your video. That might also help save a little bandwidth.
Muting the video?
Yeah, there should be a little camera. It's like a box with a triangle next to it. Just click on that.
1 second. I don't see it.
I'd be back in your Zoom screen. Do you know where your Zoom screen is?
No, I don't. 1 second. It's not working. Cancel that. You said mute video?
Yeah, we're still seeing you. Ah, there you go.
All right. Share screen. Desktop. Okay. Cancel that. Cancel.
Good, we're seeing your screen now.
Yeah, looks good.
Okay. So
Also
the payload.
vetted.
What?
Looks good. Or,
Oh, okay. The payload delivery system is comprised of the invasion device, the vesicle sensing device, and the cell lysing device. And the lastly is the vesicle lysis device. The payload device has GFP and RFP. RFP is free to spread around the cell in the cytosol. The GFP binds to something in the nucleus, which was derived from the NLS was derived from a viral system that infects the eukaryotic systems or mammalian systems. The invasion device runs on a constitutively expressed promoter. It's found in well in the E. coli that had the device, had the entire had the entire device in the genome. Oh, it relies on invasin, which binds to beta-1 integrin, and that's how it's taken up into the cell. And in cancer cells, in mammalian human cancer cells, it's especially expressed, so it's an easy target for a bacteria to be taken in. The cell responds to, what is this? I can't see this. All right.
Here, you want me to? I'll jump in for 1 second. So the idea being that the bacterial cell that's expressing this invasin will be able to bind to the host cell and then get taken up, and then it's present, and because it's taken up by it's taken up into a vacuole system, right? So the idea being after the bacteria invades the cell, it has to at some point release its contents to the mammalian cell. Right. Okay. So the vacuole, the VLD is the vacuole lysing device. I think you said that already anyway. But okay, so that's the transition that I wanted to maybe piece it a little bit. Okay, perfect. Keep going.
Okay. Various systems were tested with magnesium's stress response promoter. The stress response promoter was taken from FOPQ, a transcriptional device. FOPQ regulates homeostasis of a cell if it's missing divalent cations. That's I'm sorry. Are you seeing this slide in detail? Hello?
Yeah.
It's good enough, yeah.
Okay. Here. This is not on the paper. It's just something I found online. This is the promoter that was selected for magnesium stress response. Well, that's slide 3. That has various systems that were added one at a time. Does anyone know what the blue one is?
DNA dive.
That's generally, for, I believe, for DNA hook stains. So it's for confirmation that you're looking at the nucleus. So it's like a positive control for the GFP. So you should, if you do the merge, what is green and blue is what, yellow? Yeah. So so the hooks is just a nuclear stain.
Oh, all right.
Does anybody want to walk through those images to identify what are the cell structures that are lit up?
I'm sorry. This is the GFP of a payload device or payload generation.
Because the GFP had the nuclear binding, so that's why it matches the hook stain, because GFP is in the nucleus, but the RP is elsewhere, scattered.
So in image A, for instance, are those images or those hot spots, are they supposed to indicate that it's still in the E. coli? Is that the kind of rod shape?
Yeah, those are the only ones that aren't lysed, the only ones without the self-lysing device part. So I think that's why in that panel A, you see a much more defined localization rather than in B, C, and D, where it looks a little more diffuse.
Yeah, because that scale bar says 10 microns.
Yeah,
And so these things are like 1 to 2 microns, so that would make sense if you're bacterial or in the lysosomes.
And then B and C, it's still in the lysosome, is it?
I would.
Yeah. Yes. Yeah. Well, because it's only in D where the well, actually, so the vacuoles, not necessarily lysosome. Right? It's because the vacuole lysing device is only employed in D. Yeah.
Right, but I think is that a time scale? Let me take a look at the figure because it is getting more diffuse, right? ABC.
Well, I mean, it can still lyse the bacteria has spewed it out, but it's still in the vacuole, so it's more diffuse, but still contained back.
I like this. I think that it's well displayed, but I wish that there were some bright field microscopy so you could see.
Right, where
see where the cells are, where the host cell is.
Right.
Mm-hmm.
But to me, it looks like the trend is that it's getting lysed and you're getting a more diffuse signal. Yeah. And then after time, if you had a time lapse, I would expect it to get more punctate as for GFP would go into the nucleus as it gets started.
But only in D, because that's the only one that has the vacuole lysing device. Everything else should be maintained inside of the vacuole.
Oh, right. Yeah, so
it's pretty diffuse.
It does look diffuse, but I don't.
The vacuoles are pretty big. I don't know the vacuoles.
So then in D, the red is the entire
RFP.
This is cytoplasm, yeah.
in the entire
much more
and the
diffuse.
GFP's migrated then into the nucleus. Is that what that's depicting?
Mm-hmm. Yes. Yeah.
Thanks.
Should I continue?
Mm-hmm.
Yeah, thank you.
Oh, by the way, the A through C are in HeLa cells, and the D is in U373MG. I don't know what that is.
Oh, I didn't catch that they were different cell types. I wonder why that is.
What?
Yeah.
Did they mention, Yuriy, why there's 2 different cell types that are used?
I can barely hear you, GenSpace people.
Do they say explicitly why they use U373 MG cells versus HeLa cells for those 2 experiments that are found?
I haven't been able to find it. It's in the methods, but it doesn't mention what they are or what they do. One second.
Sometimes they do 2 different cell lines just to prove that it works on 2 different cell lines.
But it's funny that they would show
Yeah.
different.
Patrick, I had to mute you if you want to talk.
Okay. What was that?
I was just saying you had a mic open.
Okay. All right. Systems were selected for the self-lysis core and lambda-phage lytic system. Both have pollen in them. Pollen oligomerizes to puncture holes in the inner membrane. And then lysosome goes to action on the peptidoglycan layer. Does anyone understand why there was a threshold set on the whole system?
Well, I think they wanted to prevent unwanted extra lysis. So that's where that whole anti-colon, I think, comes in, but I'm not sure about the details.
So yeah, I had a question here. So what they had here with the anti-colon and the constitutive promoter, were they it was coming back. From my understanding, there was residual expression of the lysosome and olin, which very little would be very toxic to the cells. So it seems like they were getting these expressed without any promoter, right? Is that there's some expression taking place from their system, from their construct, and this was sufficient to cause lysis, so they had to have that somehow quenched.
Mm-hmm.
So in their system, did they have the antiholin then turned off?
Yeah, antiholin turns the whole thing off, and
Right, but then they
to the bottom of page 2, left side of this.
okay.
419.
Right, they mentioned that there.
internal dividend.
Because we observed that he had tightly regulated expression of folin in lysosome and well towards growth and stability, we pursued an architecture employing antiholin to prevent unwanted lysis.
Like you can't control the amount of pollen that they're making, so we're going to add an antidote rather than try to just, we have a very small amount of pollen.
Right. Yeah. So we're not that promoterless is next step. Does anybody know how the promoterless expression works?
It's at a basal leaky expression.
Right, but in their final system here, did they have a promoter driving the holin and the what's the other one? Yes, the lysozyme, and then basically overcome the antiholin's effect, or did they have basically no promoter and then just shut off with a repressor the antiholin?
I think they had a promoter, but then they found out that it's so toxic, even at a base level expression, that they removed it altogether, giving the cell a leaky expression. The leakiest of expressions.
Then they have to repress the antiholin, right? Once it gets into.
Could you repeat that?
Then they have to repress the antiholin, right, to get the system to self-lyse.
Yeah.
Okay, so since they can't control they can't control a folin, they can control the antiholin. Yeah.
That's why it's constitutively expressed. All right. The other thing that they added was bacteriocin. I think it's bacteriocin released protein that. One second. That permeabilizes the outer membrane by activating phospholipase A. That's interesting. So here's the VLD system.
Reduced, and
What's
then
that?
without magnesium. It doesn't work without magnesium. It doesn't work with magnesium, so then in the vacuum, it works because there's no magnesium. So that's how they control it. That's why they don't need a promoter, because the enzyme's not active.
I'll keep going since I didn't hear any of that. VLD is a a tricky system, I think, because DNA from the bacteria can end up. Well, it's not relevant to this paper, so therefore, Perfigolysine O is a cholesterol-dependent monomer that oligomerizes to assemble a toxic pore in a vacuole membrane. And the second module they added was the PFO, which together with PLC degrades the outer layer of the lipid bilayer leaflet by cleaving the phospho groups from the fatty acids. They use two of them. There's PLC from one species and PLC from Listeria monocytogens. That's not important. That's not important. They then upgraded from P4P promoter to a RBI. That's all I've been able to prepare. Discussions?
Yeah, so we were having a little discussion here. So just to clarify, so we mentioned this a little earlier about we were discussing the dampening of your bacterial self-lysis device. So there is a promoter actually that's responsive to low magnesium levels. So it's the lysozyme and the holicin actually get turned on even without a promoter, but they get turned on in the context of this promoter that's only responsive to low levels of magnesium. So you get this little dribbling out of holicin, which is enough. It's like really toxic to the cells. So it's enough to punch holes and kill the cells. So you've got this anti-holicin with this constitutive promoter, which whatever the levels are enough to quench that low basal level that's always coming off. And then when the cells get into those vacuoles and the magnesium levels crash, then you have this promoter that's induced by this, I believe, a phage activator, activator responsive promoter, which then drives the levels of the holicin transcripts and lysozyme transcripts up, overcoming the whatever the constitutive level of the anti-holicin is. It's kind of a seesaw, and then that's enough to induce lysis. So that's really their fix, their patch really for this leaky promoter is to have this anti-holicin kind of blanket. That's kind of a low constitutive level. Like it's overcome quickly in the context of your low magnesium inducible promoter. So,
Now, that makes sense, but is this a case of them just kind of tailoring what they got to work with the system, or is there a reason why they couldn't have picked something that has a more reliable?
They cite a reference, which is kind of interesting. I'm wondering if this is a nice common tactic, like a common patch. They cite, they cite 27, reference 27, which is Buchler Cross 2009, protein sequestration generates a flexible ultrasensitive response in the genetic net, which may have, because they meant that's in the context of using this anti-holicin to patch a leaky basal expression system. So I'm wondering if this is like a generic fix tool for synthetic biology. If instead of having a really strongly repressed promoter, you have something that acts as an antidote that sequesters protein. So you let whatever else good it is dribble out if you have enough of something that sequesters it. So it looks like that may have been in that other reference, and that's kind of a general tool to be used to tailor the system to make it fine-tuned.
In this case, based on the presence or lack of magnesium.
Yeah, in this case, tailored to the presence or lack of magnesium. Right. Anybody hear me?
Yeah, got that.
Okay.
I did have a question around 4B and C. In that case, they're charting kind of delivery. There's two things that I understand: delivery and cytotoxicity. And I wasn't quite sure. I assume the delivery is the GFP into the nucleus and the cytoplasm. But I wasn't quite sure that the cytotoxicity at this point wasn't really the end objective, was it? Is that just some sort of side effect?
I think it's based off of this, right? If the lysine wasn't sequestered, you have out of control cytotoxicity, right?
to make sure that the system isn't toxic. That's what I understand.
Yeah.
Like they want the delivery to be high and cytotoxicity to be
Pretty
low.
well, yeah.
We're talking about cytotoxicity of the Kittleson cells, right?
Or
The cell target cells, cell mammalian cell line.
Yeah, I think the combination of this delivery and cytotoxicity is just basically giving a benchmark for how reliable this lysing is happening at the right time.
So is it the VLD that's
After it didn't work. So then you can only withdraw, and it would be for other things, unless it's safe or something.
I didn't quite hear that last comment. I'm curious then, is the cytotoxicity from this the VLD then lyses the entire cell instead of just the vacuole? Or do we not know?
I think it's definitely a measure akin to that of sort of unwanted lysing throughout the cell.
VLD lyses the vacuole.
Yeah, that's what I thought. It was just the vacuole. So I guess I just didn't understand quite why that. First of all, in Figure B, the cytotoxicity was so high, and then in C, there still is a residual cytotoxicity, and it looks like it's kind of optimized, so it's pretty low.
What I like about this.
Also, look at the y-axes. Like the y-axes are quite different between B and C, percentage-wise.
Oh yeah, got it. Yep.
Yeah.
Any idea why they used PBAD, but then PBAD was never mentioned again?
It's probably a better model.
They tried to use the PBAD for the whole length.
Yeah, but did they? Is it just for modeling or for usage in the delivery system?
And I just think because PBAD is not, PBAD's pretty leaky, and you can't control it extremely easily. Like it's been shown that the PBAD promoter, even when you induce it or you like lower the concentration of arabinose to induce, you're not really lowering induction, but you're just turning, some cells are not on and some cells are on. So you get this mixed population. So I just think the PBAD promoter is not stable as a whole. So, to explore a better regulated system besides that was most likely the intent.
So I don't want to ask too many questions, but if nobody else has one, I'll jump in with another.
Keep asking.
Well, so the other one was I wanted to kind of understand the cell selectivity of this mechanism, of this device.
You mean the use of integrins?
Yes.
That there really isn't a cell selectivity?
Okay.
Is that what you're getting at? Yeah, there really isn't one.
Okay.
Yeah.
So to be an effective therapy, then there's some other invasive device that they need to come up with then. It's going to be more similar.
They have. In supporting materials, they mention methods, and they added, I think, one more device to it to be a better carrier in the blood system. Let me find it.
I didn't read the supporting parts, Yuriy. What did they say?
1 second.
It wasn't not really so much about carrying, it's about targeting.
Here on the last set of the methods, the biosafety and biosecurity does say that they made it sensitive. So it's kind of like a Jurassic Park thing where, like, you know, the Jurassic Park can't breathe. So they made it like iron sensitive to make sure like it won't survive, like it made that. Yeah. So that's kind of smart. But I don't think that this is a selective.
Or maybe
You know what happens in Jurassic Park.
They mutated. But that said, I don't think the invasion like this could be possible. I don't think it's a good.
Or maybe that's not really
target.
part. Maybe that's not really part of their system because ideally something like this should be used for any point targeting, right? So maybe that's not
Yeah,
the
so it
core.
would be changed.
It's
Yeah.
really not the important part of what they do.
Also, I don't think bacteria target cancer specifically. So we have to clear that because we don't have.
But there are markers for cancer or things that are overexpressed, and I think they're arguing that in this one reference, in this reference it says beta integrins are overexpressed.
Yeah, we have to find something, but then you have to
Like more
that's
selective.
overexpressed, and then you have to find something that's bacteria, or then you have to.
Do they not have any other young animals that's go? That's maybe something.
It gets expressed in most of the cells. That makes sense. Right.
I'm saying, when the bacterial lysis and it still that, their payload, but isn't also spinning out like the bacterial genome and stuff like.
Yeah, it is, but usually that cell itself is going to be targeted for expression. It's going to be expressed as cytotoxic or block. But you don't want to
The reference for, because I had meant to look this up, the reference for beta integrins being overexpressed on human cancers is actually about how beta integrins induce bacterial uptake. It's not about cancer.
Mm-hmm.
I really didn't hear anything that you guys said on. I'm having a hard time hearing everyone at Gen Space, so I've been missing quite a bit. I apologize.
Audio.
Maybe somebody could summarize.
Yeah.
Julie just said that she looked up the reference for beta integrins, and it wasn't particular to cancer. It was a paper on the
uptake, which is the second reason why they chose it, because something that binds to integrin will induce phagocytosis. And so it can get into the cell.
Yeah, but I also think like even if the target specificity to cancer cells, like I don't think that's really necessarily what's important here because this platform that they're trying to build should be sort of like target agnostic that can be used for any kind of targeting mechanism. So I don't think they're really caring too much about that part. So they're just using something that's going to be uptaken into the cells really easily so they can show that the system works.
Mm-hmm.
Correct, yeah. More for broader gene therapy purposes. I think it would be nice if we could start building some devices that, like, if cell infected with X virus, then kill, kind of a thing. Or, there's a whole range of things that you could potentially use a technology like this for, assuming we can build the correct devices. But
I don't know. Maybe this is just me, but I think I would be more comfortable with this system we could really somehow stop make sure that these cells don't replicate. The E. coli. Yeah, we have a system here that basically is capable of very efficiently entering any human cell, right? And if your cells don't auto-destruct, then what? You have something that's like listeria, right? Which is a known human pathogen that basically docks to, adherents and enters cells, which is, a cell surface protein, and, that's a known human pathogen. So, you're coming up with a toolbox here, giving it to a bacteria that essentially allowing you with extremely high efficiency to enter any human cell. And you're relying on a mechanism for the cell to lyse, right? Personally, I would feel more comfortable if you had a deactivated, completely replication deactivated system, like a bacterial version of like the telomerase, negative cell. Though, of course, bacteria don't have telomeres, but I'm saying something that basically is really, maybe even a synthetic system using lipid vesicles, but not something that's intrinsically self-replicating. Because essentially, there is cytotoxicity here, right? I mean, cells don't lyse, and then what? They're just replicating, and they're once they replicate and they exit the cell, they're just going to reinfect other cells, right? I mean, I don't know. I know that's just like an issue I had, and I know there was a team in IGEM.
At the very end, sorry, to address Oliver's point about safety, it says at the very end, biosafety and biosecurity considerations in the methods. To mitigate potential dual use, only RG1 parts are fully described. You have to show institutional approval from your institution, I guess, before you will be given full access to the information. That's interesting.
Until what? You get access to the full information?
Yeah.
I mean, it kind of sounds like it's kind of fairly straightforward, right? I mean, you have an invasive protein which is well characterized for a long time, being expressed that binds to a beta integrant. So, I don't know how much else there is to it.
I'm just saying they also considered that maybe there would be a bad purpose for this or a negative consequence, I guess.
Yeah. I mean, they do mention that, but I'm just kind of guessing it might not. It would be good if the system was just not capable of replicating. Period. If this was a system where you had it expressed in, like even a viral delivery system that is completely devoid of any kind of autonomously replicating system, right? Because it is kind of that what if, right? If that one module doesn't work, okay, then what's the option? The option is uncontrollable replication and lysis of all cells, right? So clearly you don't want that option. so and there doesn't appear to be a backup for that option, is what I'm saying.
Backup could be antibiotic sensitive.
Right, okay, an antibiotic. So if it's an antibiotic sensitive strain, right? So in all things considered, these are E. coli MG1655. These are K12. So these are like as benign as you can possibly get. So these things will die if you sneeze at them or give any kind of antibiotic, even the weakest antibiotic. So it's not so much of a concern with this strain, but the potential is there. Since it is a self-replicating, system that if you don't have that barrier to auto-lyse, then and you don't have, let's say, antibiotics, then the cell is basically replicating.
They could go with something simpler. Some insect gut bacteria, they have a very minimal genome, and those could be really, really weak compared to, let's say, Listeria or even E. coli.
Well, they will use Listeria.
Yeah, I mean, there could be other methods like employing some sort of auxotrophy or the reality is, though, this project was actually started under, I believe, under the tumor-killing bacteria testbed that employed by Simberg. And so being the idea that there would be an engineered device within bacteria to kill tumors specifically. And this project was moved from cell culture into mouse models, and then the project stopped about a year or a year and a half ago, at least in our labs.
What do you mean by stop?
Zombie outbreak.
Like, it is no longer being worked on in our lab.
Was there any data from the mouse experiments?
There is some data somewhere that I could probably dig up. A graduate student that left about a year plus ago. I was finishing up the project, and after doing some preliminary in vivo studies, it just was. I think I don't quote me on this because I really wasn't involved in this project at all, so I don't remember the results. But I believe there was something like they had efficient uptake maybe in culture, but not in whole animal. And I don't remember if they were doing systemic injections or direct injections into tumors in the animals. And I don't know how much troubleshooting actually went into it beyond what was done in culture. I don't really know how much troubleshooting to get something to work in a animal model was actually done. But I can try to look up that information and pass it along if
could you post the supplementary information? I see Yuri's got some supplemental discussion up there. Looks very interesting, where there's actually an intravenous delivery to a mouse.
Oh, so maybe it was so maybe they did include it. Okay, let me see.
Yes, please.
That's interesting because these are they're intravenous delivery, and so now we're going to in vivo models with these E. coli MG155 strains. I mean, they have lipopolysaccharides, right? So are these special mice and they don't have like some toxic? Because I would expect there to be like an immune response, like the mice go into shock if you're.
How would they administer it? Like putting it in the bloodstream would be different than giving it orally versus topically.
Yeah. Although I did read somewhere that mice, at least wild-type mice, have a very high tolerance for sepsis, for having bacteria floating around in the bloodstream. So unlike humans, which you would have, you go into shock if you were to deliver, kilos of E. coli into your bloodstream. So I don't know. Maybe that works well with the mouse model because they have a low shock response and high sepsis threshold. I don't know. Does anybody want to comment on that? Or are we getting too off the topic of the paper, getting into the in vivo mouse models?
No, we're not. I missed a little bit. Are you saying that there's a difference between male and female mice, or did I completely mishear that?
No, I said that. Oh yeah, there is.
Fundamentally, yes, but okay.
Well, I was just saying that with mouse in mice models. So first, I was curious as to how to deliver it, and we were discussing whether it was injected or some kind of topical system. But because obviously injecting E. coli into humans, you would get a shock response because of lipopolysaccharides, and your immune system would respond to it. And then I remember reading somewhere that mice, at least wild-type mice, tend to be highly resistant to sepsis. They can tolerate actually quite a lot of bacteria in their bloodstream, which maybe I'm mistaken, but I'm pretty sure I read that in a report. So maybe that system works well in a mouse model, wouldn't work in another mammalian system. Actually injecting E. coli and MG155 into a mouse and getting not getting any like lethality from a shock from the immune system. So I don't know. If that's what they did with this in vivo system, they just went directly and injected it.
I think you can coat bacteria with PEG, maybe. We can look into that to avoid any sort of immune response, or to like lessen the immune response or maybe evade the immune response. And I don't know, hold on, I'm looking it up. I think PEG has been shown to, PEG coating bacteria have been shown to prevent immune recognition, but I'm trying to find that. But yes, I would agree with you. I think there people are looking into ways around that. Right now, I don't know how much research is being done with using bacteria in mammalian systems anymore. And I want to say there was also quite a bit of kickback with the gene therapy woes and in the synthetic biology community. So right now, focus has shifted quite a bit. There are some labs doing mammalian systems, but I think they're exploring other avenues for delivery, such like viruses and other nanoparticles. But
What gene therapy was?
What gene therapy was?
Yeah.
I think just the concerns that people have with not being able to specifically target cells efficiently yet, like certain populations of cells. And it's getting better. People are modifying nanoparticles, whether it's a virus or other capsule, to potentially target specific tissue types, but there's a lot of worry there that you get off-target effects, and how do you modulate that? That's a fairly large concern still. So things that are like systemic disease issues are better targets, like when you want to target every cell. So you're not necessarily as worried. I think one of the reasons why this is getting totally off topic now. I think one of the reasons why muscular dystrophy has been an interesting target for those in gene therapy is because when you deliver that IV, you want it to get into like the cardiac muscle and the smooth muscle tissue, and you kind of want it to go everywhere. But anyway, I think synthetic biology is kind of crossing that path. I think synthetic biology is kind of crossing into that territory right now because it's at its infancy, and people are trying to think of broader applications. And now, it's getting to the point where, engineering mammalian systems, that's an interesting topic. And there is potentially more room to play, because if you can build all these different sensing devices and have things turn on and off in certain scenarios and like lead to this cascade of events, I mean, there are kind of like these endless possibilities with what you could do in a mammalian system. So where gene therapy was before, with maybe just delivering like a single gene to replace a mutant gene or something like this, now we're dealing with these like more advanced complex systems, and making sure they function properly, and being able to deliver these large things efficiently, and sense the right on and off molecules and things like that. I think that poses a whole other challenge to the community. But that's just my assessment of it, though. I don't know if anyone wants to weigh in there. But I find this area the most interesting. I think, I'd like to see synthetic biology cross over into this domain more. But
Nina, do you know, are there any viruses that are being used for that kind of synthetic engineering of the pathways or devices or whatever? Because I assume it's got to be a pretty big virus to carry a whole payload like that.
Yeah, like adeno-associated virus has a small packaging capacity as opposed to like adenovirus. And there are some other systems as well. But I would say if you want to, the two labs that I know for a fact that are working in mammalian systems right now are Ron Weiss's lab, and Ron Weiss is at MIT or Harvard. Don't quote me on this. Hold on. I will tell you momentarily. Ron Weiss is at MIT, and also Pam Silver, and she's at Harvard. So I would look at the research going on in those two, because right now I think those are the only two labs I know of that are in synthetic biology or systems biology working in mammalian systems. Yeah.
There was also a mammalian system, just to throw another name. There was a paper, first author Lev Scott.
Oh.
It was a there's a light-inducible gene regulation system that was actually employed in mammalian cells. If you Google Lev Scott, it's I don't know where I can't remember which lab. It was a postdoc in, but they were doing synthetic biology primarily in mammalian systems.
What was the name?
LEBSKAY. So it's the Voigt Lab, Christopher Voigt.
Ah, okay.
And Andrew Ellington. So they basically did the bacterial photography, but I think they employed the system later in mammalian cells to turn on proteins. And I don't know which mammalian system they used, whether it's I think it's a mouse cell one. So I don't know what they're doing now with the system.
What's happening? All right. Does anyone have any other questions, or I don't know. I think there is room for DIYbio folks to get involved in building devices like this. I think it's interesting. Go ahead.
No, I was about to say I like some of the obviously the whole key is to take the modularity of the system and you test all of these modules and kind of breaking it down into ways you can and I just kind of for some reason I zoomed in on this fix or this hack where they use to quench the leaky promoter, and it seems like that's a general system, right? So I don't know if that's, something like this you would expect nature probably already uses. But it'd be interesting to compile an assortment of these engineering hacks that, a list of, what are all the ways you can fix a leaky promoter? Well, you can have, a repressor. You can have a system that binds to and deactivates the protein that leaks out, right? And measure all of these, the effectiveness of all of these solutions. So this would be like a subsystem, right? And then the subsystem would have all of these. So you can have either a really weak promoter, or you can have these fixes that even though the promoter is leaky, quench it to zero. So that's always, as a biologist, that's always been the problem, right? Because systems are sloppy. They're always, you get some leakiness. So the effect is always, some to a lot, right? So it's never from 0 to 1. It's this kind of threshold, but they seem to have gotten around that here. So this is a really, and they really quenched it to 0. So that really has made the system, at least in this respect, truly digital, right? So it wasn't slightly on, because in this case it would be toxic, and then on a little more, like you would see in a proof of principle scientific experiment, little GFP, a lot of GFP, okay, so that's having an effect. But in this case, since they're engineering something, even a little is not acceptable. So they had this system that was fully off, then on, and it got an effect. So I thought that was, to me, that little part was really cool. And it'd be nice to see that kind of, I'm sure it's going to be expanded upon further as in synthetic biology. We're all passing the torch, so to speak, to the engineers and software guys, right? So all this.
Mm-hmm.
I have to say one thing though. I think it's funny that when they put it with a vacuole design, they still had to write another patch though, which is they had to do a second fix. When they put it on the vacuole, they wouldn't rise completely because they probably still have calcium. So then they had to add something else. So I think that kind of speaks to you have to, when you make it more complex, there's unexpected problems that arise, and they had to do a second patch, which is fine, but it's 2 and 2 is not 4 biology.
Right. So if you're designing an in silico system, you probably need to have that already embedded. So if you just design it in silico, the software can tell you the algorithm. Here are all the potential problems, and here are the patches for it. So you might have to make your system, that's how you.
I think they mentioned that as a path going forward at some point in silico design. They mentioned somewhere. The bottom of page 422 on the left side, right for the methods, we talk about that and how that would be an ideal going forward.
Right.
Yeah. Cool. So then we can build that computer software for component design.
Yeah.
Yeah.
Did anybody catch when they were assembling the independent devices? Did they like assemble those all on a single plasmid or just a bunch of plasmids, or how they did that?
Yeah, that's a good question.
We do talk about it. I don't remember where.
Because the vacuole sensing, not the vacuole sensing device, because the payload generator, right? That has to be on it, right? Because that has to be is that the only system that's expressed off of a eukaryotic promoter? Because that plasma has to be expressing the GFP and the RFP, right? That's after it's already lysed. So I'm assuming that has to be a eukaryotic promoter. So is that a separate? Is the payload generator with those signals, GFP, RFP proteins? Is that a separate system? Because self-license, vacuole sensing, vacuole license device, and basin. I mean, those all could be expressed within the E. coli, and then it all explodes. And then you still have to have the GFP and RFP expressed. So that ought to be from a eukaryotic and a separate vector, right? There it is, right? Constructed using Babel break standard assembly, BioBreak assembly.
I mean, it looks like the GFP is expressed in the bacteria prior to lysis, right?
Yeah. So it's all from. So then enough of it is.
Yeah, I guess there's just enough that when it lyses can find its way to the nucleus.
Yeah, in fact, we know that right from those images because they were, the GFP was before lysine. You could see the GFP in the image, A or whatever it is.
Yeah, right.
Yeah, in the bacteria itself.
But then, okay. That makes sense, but does it, I guess, how long are these images incubated for? Because whatever GFP you have there, right, because it's got once you have that lysis occur, then that's it. Whatever GFP and RFP made is it. So it's got a half-life. And I don't remember off the top of my head what the half-life of GFP and RFP is. Like they have extended ones, ones that don't get degraded, proteolytically degraded. So I don't know what the half-life of these GFPs and RFPs is in the context of this experiment before the signal goes away. Because if that's true, then it's not being expressed anymore. That's the GFP that just gets delivered into the cells. I don't know if they mentioned that somewhere here. On how long these images were incubated for.
And let's see, in the results it says, what did it say? For the 3 to 4 hours. I think cells were infected and incubated for 3.5 hours before microscopy. Is that right?
It's about right. I mean, if it's got an NLS, we'll probably get within the nucleus within minutes upon lysis. It's actually pretty quick, at least in yeast.
I think what's on each plasmid.
Okay. So yeah,
Sorry, I didn't mean to cut you off.
No, that's pretty much all.
I think the plasmid information is in a supplementary table. Oh my God, I can't talk. Table 2. So once we post that, hopefully that'll answer that question. I think maybe Yuriy, did you send that link out?
Yeah, it's in the chat.
Okay, perfect. Thank you.
Sure.
All right. Are there any more questions?
And
So I guess for the 19th when Chris is present, should we just via email come up with a list of questions, or someone do a little bit of documenting? Yuri, I don't know if you were documenting our conversation a little bit, or we can just go back and rewatch our recording.
Yeah. Yeah. I mean, I think we need if we all come up with a list of questions. I think each of us has a different kind of area of concern or some some different facet that we're interested in.
Mm-hmm.
I mean, I haven't obviously read all of his papers, but
Yeah, is the discussion supposed to center on this paper or synthetic biology? Is there a theme?
I think you can ask whatever question you'd like, including about his career track or anything like that.
So you want to keep it really open-ended and potentially going everywhere from specifics onto a particular paper like this recent one here to, I don't know, advice to a young investigator. I mean, I'm comfortable with that. I mean, if Chris is comfortable, I mean, it's up to him, right? I mean, since he's going to be.
Yeah, the whole thing will be curated by his administrative assistant anyway. The whole list of questions.
What do you mean curated?
I guess he'll put them in order of importance. I'm not sure.
Oh, so you want to send him questions before the meeting?
Sure, yeah.
Is that what the idea is?
I think we should probably start to share questions in the group, because that way, everyone has the same question. It won't be sent 5 times.
Yeah.
Mm-hmm.
Okay. So how are we going to do this? Are we going to just have an audience and the curator is going to ask questions? Or
I don't know. In this part, I didn't understand before.
Because last time I think we had it when we had our last lab meeting, it was going to be sort of open. Like we basically have Chris join and people. We all share the questions. That way, somebody might already have the same question, and then everybody joins the meeting, and you if it's your question, you ask the question. Or a modification thereof if you thought of a modification. I don't know, how does that work? I mean, because it is a small enough.
Maybe we could ask him what?
I'm just saying it's a small enough group. It's not like there's going to be 500 people in attendance and a microphone has to be passed around.
Yeah. Maybe we could ask him questions after after he runs out of questions to answer. Open-endedly.
So like if we think of anything during the discussion or something.
I guess I'm still a little confused on the format. Like, did he want us to send him questions ahead of time and then he's just going to run through the list? Or did we? I don't know what was.
I'll forward you the email.
Okay, perfect. That sounds perfect. Okay.
Great.
So no paper for next time, just a discussion.
Yeah, and it's the 19th.
So next Tuesday. Tuesday, 19th. 19th Tuesday.
Okay, well.
I probably will not be able to attend because I have a class starting at 9 o'clock. So I will at least miss a half hour, which is probably, But maybe I can forward the questions.
Okay.
And it is YouTube. It's okay.
I could actually maybe have my class go to the discussion.
Oh.
It's only 4 students.
Yeah, you should just bring your class to Journal Club.
Yeah, I should. And they're engineers, so they'll get a kick out of this.
Perfect.
Works.
Okay, excellent. So then I could do this from Cooper Union while everybody does from here. Okay, sounds good to me. I think that's planned.
Okay, cool. All right. Well, I guess then people will just email questions to the list and we'll compile.
We should probably have a cutoff date. How about like the 17th, that Sunday night?
Okay.
Sounds good to me.
Sunday night it is.
So I think this is the lowest energy wind down we've had yet.
Yeah.
I'm going to stop the recording then.
It's daylight saving.
That's a, you're editing this, right? So we can get those awkward pauses out of the.
No, they're all there.
They're all there.
It's
This is being live streamed to Times Square.
See you later.